Chapter 1. Introduction to Clinical Trials. The evolution of the modern clinical trial dates back to the 18th century. Lind, in his classical study on board the Salisbury, evaluated 6 treatments for scurvy in 12 patients. One of the 2 who was given oranges and lemons recovered quickly and was fit for duty after 6 days. The second was the best recovered of the others and was assigned the role of nurse to the remaining 10 patients. Several other comparative studies were also conducted in the 18th and 19th centuries. The comparison groups comprised literature controls, other historical controls, and concurrent controls. The concept of randomization was first introduced by Fisher and applied in agricultural research in 1926. The first clinical trial that used a form of random assignment of participants to study groups was reported in 1931 by Amberson, McMahon, and Pinner. After careful matching of 24 patients with pulmonary tuberculosis into comparable groups of 12 each, a flip of a coin determined which group received synocresin, a gold compound commonly used at that time. The British Medical Research Council trial of streptomycin in patients with tuberculosis, reported in 1948, was the first to use random numbers in the allocation of individual participants to experimental and control groups. The principle of blindness was also introduced in the trial by Amberson and his colleagues. The participants were not aware of whether they received intravenous injections of synocresin or distilled water. In a trial of cold vaccines in 1938, Deal and co-workers referred to the saline solution given to the subjects in the control group as a placebo. One of the early trials from the National Cancer Institute of the National Institutes of Health in 1960 randomly assigned patients with leukemia to either 6-azarocil or placebo. No treatment benefit was observed in this double-blind trial. It is only in the past several decades that the clinical trial has emerged as the preferred method in the evaluation of medical interventions. Techniques of implementation and special methods of analysis have been developed during this period. Many of the principles have their origins in work by Hill. The authors of this book have spent their careers at the U.S. National Institutes of Health and Academia. Therefore, many of the examples reflect this experience. We also cite papers which review the history of clinical trials development at the NIH. The purpose of this chapter is to define clinical trials, review the need for them, and discuss timing and phasing of clinical trials. Fundamental point. A properly planned and executed clinical trial is a powerful experimental technique for assessing the effectiveness of an intervention. What is a clinical trial? We define a clinical trial as a prospective study comparing the effect and value of interventions against a control in human beings. Note that a clinical trial is prospective rather than retrospective. Study participants must be followed forward in time. They need not all be followed from an identical calendar date. In fact, this will occur only rarely. Each participant, however, must be followed from a well-defined point in time, which becomes time zero or baseline for the study. This contrasts with the case control study, a type of retrospective observational study in which participants are selected on the basis of presence or absence of an event or condition of interest. By definition, such a study is not a clinical trial. People can also be identified from hospital records or other data sources, and subsequent records can be assessed for evidence of new events. This is not considered to be a clinical trial, since the participants are not directly observed from the moment of initiation of the study, and at least some of the follow-up data are retrospective. A clinical trial must employ one or more intervention techniques. These may be single or combinations of diagnostic, preventive, or therapeutic drugs, biologics, devices, regimens, or procedures. Intervention techniques should be applied to participants in a standard fashion in an effort to change some aspect. Follow-up of people over a period of time without active intervention may measure the natural history of a disease process, but it does not constitute a clinical trial. Without active intervention, the study is observational because no experiment is being performed. Early phase studies may be controlled or uncontrolled. Although common terminology refers to Phase 1 and Phase 2 trials, because they are sometimes uncontrolled, we will refer to them as clinical studies. A trial, using our definition, contains a control group against which the intervention group is compared. At baseline, the control group must be sufficiently similar in relevant respects to the intervention group in order that differences in outcome may reasonably be attributed to the action of the intervention. Methods for obtaining an appropriate control group are discussed in a later chapter. Most often, a new intervention is compared with, or used along with, best current standard therapy, only if no